Hello, everyone. In this chapter, we are going to address the pattern of chromosome inheritance. As we address this chapter, I want you to focus on a few things. One, it's what is the structure of chromosomes? Two, what is the cell cycle and what happens during the different stages of the cell cycle? I want you to be able to explain the two division of meiosis, but also the segments of mitosis, which is the second type of cell division. I want you to be able to compare what happens in meiosis versus what happens in mitosis. That way, you are going to know not only what is happening in the two types of cell divisions in our body, but also when they apply and where they apply. Now, during cell division, things do not always go as planned. There are some events, including a particular type of event that we call non-disjunction. I would like you to know what non-disjunction event is, because this is going to allow us to address situations when gametes have either too many chromosomes or too little amount of chromosomes, right? And also, what are some syndromes associated with unequal distributions of chromosomes during the formation of the gametes? Now, when we talk about the formation of a gametes, where and when do they happen in males and in females? And also, we are going to complete this chapter by looking into the changes that occur in the structure of a chromosome because of some accident that can happen as the cell divides. With that being said, as we are going into this, I would like you to walk in with a number of knowledge that we already covered, actually aspects that we already covered. One, I want you to mark a pause. I want you to think about what is DNA? What is the structure of DNA? What is chromatin? Right? What is chromatin? And also, what is the structure, the complex structure of chromatin as DNA mingles with proteins? Finally, I want you to focus on the role of microtubules in the cell and the role of actin filaments in the cell. So reviewing that is going to get you ready for this chapter. With that being said, hopefully you have reviewed on this topic, and now let's dive into the structure of the chromosome. Now, when we consider the chromosomes, we can wonder, okay, what are chromosomes? Well, chromosomes are basically DNA and proteins condensed together to form a visible structure when we look at it under a microscope, right? Now, obviously, we cannot observe chromosomes distinctly under a microscope at any stage of the cell cycle. That is not going to happen, right? Most of the time, though, chromosomes are going to be in the cell in the form of chromatin. Chromatin, it's DNA and proteins The particular proteins we are talking about are histones. Let me write it down. Histones. The histones are going to mingle with DNA to form a diffuse structure in the nucleus called chromatin. That diffuse structure, it's in a state where genes that are actually part of the chromosomes, right? Genes are carried on the chromosomes. Genes are going to be able to express when the genetic information is organized in terms of chromatin. And again, chromatin, it's basically DNA mingled with proteins called histones, and that DNA is relatively loose, which is compatible with gene expression. But when that DNA mingled with protein is very compact and visible under a microscope, we call that chromosome, and at that point, most of the genes are not readily available for gene expression. Let me close that parenthesis. Now, in a human cell, most of the cell in your body are going to contain 23 pairs of chromosomes. These 23 pairs of chromosomes are organized in what we call homologous chromosomes. In other words, when you were born out of a sperm that came from dad and an egg that came from mom. The egg contained 23 chromosomes. The sperm also contained 23 chromosomes. Each of the chromosomes in the sperm and the egg has its equivalent so that they are organized by pair, right? You have pair number 1, pair number 2, pair number 3, pair number 4, and so on until pair number 22. However, you have one pair, the 23rd pair, that may be homologous, may not be homologous, right? Let me address that a little bit here. Now, the 22 first pairs of chromosomes have nothing to do with your sex attribute. These 22 pairs of chromosomes are called autosomes. The autosomes are not involved in determining if you are a male or you are a female. No, they are not. Now, the one pair that determines if you are male or female are going to be called sex chromosomes. These sex chromosomes occur in two kinds. You have chromosome X and chromosome Y. For a male in a humankind, a male carries a chromosome X and a chromosome Y. A female in the humankind is going to carry one X and one X, so that's two X's. All right? Now, I'm not going to go into the intricacies of what specifically exists in the Y chromosome to determine that someone is going to become a male versus becoming a female. That's another question that I'm not going to dive in. Now, as we consider the cells in our body, we have two major kind of cells based on the amount of genetic information these cells will have. We have what we call somatic cells, okay? Somatic cells. And we have what we call gametes. Gametes or sex cells. or reproductive cells. Reproductive cells. Now, what is the difference? The somatic cells are cells of the body. These cells of the body, some examples are your skin cells, your liver cells, your nerve cells, your muscle cells, your kidney cells, and so on and so forth. These cells actually contain the two types of chromosome you receive from mom and from dad. Altogether, they contain 46 chromosomes. These 46 chromosomes being in reality 23 pairs of chromosomes. So 2 times n, 2 times 23, it's 46, right? In the humankind, n equals 23, okay? Because they occur in pair, we say 2n, right? Now, these somatic cells are diploid because they contain twice the amount of chromosomes that reproductive cells contain. Now, on the other hand, reproductive cells or sex cells, or also called gametes, contain only half of the number of chromosomes found in somatic cells. They contain 23 chromosomes. A sperm contains 23 chromosomes. An egg contains 23 chromosomes. Obviously, these chromosomes are unpaired, right? They are unpaired. And because we know that n in human is 23, right? So I want you to remember that n in human is 23, right? Now, because gametes only contain half of the amount of chromosome contained in somatic cells, these cells are said to be haploid. I want you to remember that. So we have really two kinds of cells. Cells that are diploid and cells that are haploid, right? So now, there are some techniques we can use to determine not only the amount of genetic information a particular individual has, but also the type of genetic information that an individual has. Now, I'm going to explain a little bit. These days, because of the need to prevent some genetically transmitted diseases, congenital conditions, these days, parents, mom and dads, when they are expecting, they are curious to know what the genetic makeup of their future child is going to be. And this is particularly true with the cases where there is history of major genetic diseases in the families. Now, there is this thing in biology that we call a karyotype. A karyotype, it's a chart. That chart lays out the 46 chromosomes. One can obtain a cell from the skin, a cell from the cheeks, a cell from the liver, the muscles, or a cells from white blood cells. And out of these cells, typically, the cells are going to be cultured in a lab, and DNA is going to be extracted. As DNA is extracted, this DNA is stained. Because of the stains, the chromosomes are going to stand out, right? Some areas are going to be dark, other areas are going to be clear, right? Or light, let's put it that way, right? And at that point, we are going to organize the chromosomes by size, typically from the longest two pairs of chromosomes to the shortest pairs of chromosomes, so that the pair number 1, it's made up of a set of homologous chromosomes, one coming from mom and one coming from dad. Pair number 2, made up of also a set of homologous chromosomes, one coming from mom, one coming from dad. And what makes them homologous? It's because they don't only have the same shape and the same size, at least in terms of similarity, right? But also they contain the same genes. So say we have here on the chromosome from dad a gene that we're going to call gene A. Now, on the chromosome we receive from mom, we're going to have at the same location a gene that we call gene A. Now, are these two that we receive from mom and dad exactly 100% identical copies or not? That's another story, right? But what we know is that in homologous chromosomes, when you have a gene called A from mom, you're going to have a gene called A from dad. That's what makes them homologous chromosomes. And you are going to see that on a karyotype, right? The first homologous chromosomes are the longest, and the 22nd homologous chromosomes are the shortest, right? So they are organized by size, but also by shape. And using that chart, the karyotype, we can tell if an individual has indeed 46 chromosomes or not, right? This karyotype we have here is the karyotype of a male, right? Because we have the 22 autosomes, so 1 through 22 pairs of autosomes. These pairs of autosomes are chromosomes that do not determine your sex, whether you are male or female. They are not involved in that, right? These are the autosomes. And then you have a pair of sex chromosomes here. We can tell this is a male because we have an X and a Y. Notice that the X and Y are not rigorously homologous here. Why? Why can we say that? Because they don't have the same size. They don't have the same shape. Obviously, they are not going to have the same content in terms of genes, right? So that is a major difference. However, we are still going to say that X and Y are homologous chromosomes for their function, right? They determine the sex of an individual. That is the only reason why we are going to classify them as homologous chromosomes. However, in a female that has two X's, right? A female is going to have perfectly homologous chromosomes, so that in a female, the 23 pairs of homologous chromosomes are indeed homologous chromosomes, right? While in a male, 22 pair are rigorously homologous chromosomes, while one pair, the sex chromosomes, are only homologous by their function. With that being said, let me also mention something. One thing that is characteristic to a karyotype, it's that oftentimes to do a karyotype, cells are going to be harvested for analysis after they have undergone replication. Replication is a process by which the chromosome that you receive from mom and the chromosome you receive from dad have been copied for cell division, right? That is the very reason why if we focus, for instance, on pair of homologous chromosome number 5, right? When we look at the graphic representation, we see that the pair number 5 is actually organized in such that, let's say that this is the one you receive from mom and the one you receive from dad, each of the chromosome has been copied in such way that each of the chromosome is now 2, right? Each leg in this duplicated chromosome is called sister chromatids. And the sister chromatids are held together at the particular area of the chromosome that we call the centromere. This is the centromere, right? The centromere is the structure that holds the sister chromatids together, right? So that's something you want to know. All right, let's move on. The next step is going to be the cell cycle. The cell cycle, it's basically all the events that occur to a cell from the time it is developed to the time it produces 2 brand new cells. That cell cycle is going to be divided into 2 major stages. We have a stage that we call the interphase and a stage that we call the M phase or mitosis, right? The interphase, as you can see in this graphic representation, is the longest phase during the stage of the cell cycle, right? That phase actually lasts about 90% of the time of a cell during the cell cycle, while the mitotic phase, the M phase, lasts about 10% of the time. Now, during interphase, there are a number of events that are going to happen. And interphase itself is going to have subphases. Now, a brand new cell is going to go through a subphase called G1. That G1 phase, it's a phase during which the cell is growing, right? It is growing. It is doing the job it was designed to do in the body, right? Then we have another phase. That phase is the S phase. The S phase, S stands for synthesis. But synthesis of what? Synthesis of DNA. So before the cell divides, it has to copy its genetic information through a process that we call replication. That replication is what guarantees that the future daughter cells are going to have the exact same amount of genetic information, right? Now, not only DNA replication happens during the S phase, but you also have a continual growth, right? Now, for a new cell to develop, that new cell will have to inherit all the cellular content, including DNA and all the organelles. When I say organelles, I mean, for instance, the Golgi apparatus, the endoplasmic reticulum, the lysosomes, the peroxisomes, the what else can I mention? The rough and smooth endoplasmic reticulum, the mitochondria, the cytoskeleton, and so on and so forth. So all this cellular structure have to be present in all the 2 new daughter cells, right? For that to happen, the cell has to have enough cytoplasm to distribute among the 2 new daughter cells, right? So then, after synthesis of DNA, assuming that everything went right and that the cell commits for cell division, That cell is going to enter another stage of interphase that is called G2. During that G2 phase, the cell will keep growing, but very importantly, the cell is going to prepare all the material needed to enter the mitotic phase. And that mitotic phase, it's actually the division of the nucleus. Okay? That division of the nucleus is going to happen into 4 subphases. We are going to have prophase, metaphase, anaphase, and telophase. Now, you see that between prophase and metaphase, there is a phase that we call prometaphase. That phase is actually an intermediate phase where the cell is fine-tuning everything that is necessary so that the distribution of the chromosomes is exactly happening in such a way that the 2 new daughter cells are going to contain the exact same amount of genetic information. All right? This is crucial for the cell to keep moving forward and also for the cell to survive, right? Finally, but not least, during the M phase, beside distributing the genetic information, there is also going to be distribution of the cytoplasm. That distribution of a cytoplasm carries a specific name. We call this cytokinesis. Cytokinesis, it's the division of the cytoplasm, the distribution of the cytoplasm among the future 2 new daughter cells. Very important to remember. Now, assuming that we have too many cells or cells that are damaged, well, at that point, the body is going to decide of the fate of that cell. That cell can undergo what we call programmed cell death. That programmed cell death, it's basically what I call a clean death. It's basically a cell committing suicide because there is something deeply wrong. Something deeply wrong can be, for instance, DNA that is too much damaged to be passed down, right? It can be unequal distribution of the genetic information, too bad to be passed down, right? So anything that can compromise integrity and the function of the cell, this is going to trigger programmed cell death or apoptosis. I want you to remember that term, apoptosis. Now, after a new cell, 2 new cells have developed, sometimes in our body, some cells, as they reach the G1 phase, rather than keep moving toward the S phase, they can halt in the G1 phase and spend most of their time in the G1 phase. At that point, we don't call that G1 phase anymore G1 phase. We call it G0. This happens typically for cells that take a long time to divide. For instance, muscle cells and nerve cells, to name only these 2, right? Now, to get you to kind of appreciate what I'm saying, imagine you have a cut on your skin, not a deep cut, right? That cut takes few days to repair. But on the other hand, if you have a muscle injury, that muscle injury takes forever to repair. Why? Because the muscle cells take long to repair. Why do they take long to repair? Because after a muscle cell divides, it enters a stage that we call G0. So now, for that cell to leave G0 to undergo now the S phase, it's a process. Okay? It's a process. That is not a simple process, but it is a process, right? I want you to remember that. All right, so let's move on. So the cell cycle, it's two-part, interphase and M phase, cell division or mitosis. Okay, rigorously, mitosis, which is a type of cell division, it's a division of the nucleus. Why? Because for a long time, during cell division, cell biologists only focused on what was happening with the genetic information. And because the genetic information is enclosed in the nucleus, well, they were only paying attention to what was happening with the nucleus. That is the reason why cell division of body cells or somatic cells is called mitosis. That is the reason why. Now you know. So going back to interphase, we have the G1 stage, right? The cell doubles its organelle and the cell grows in size, does what it's supposed to do. The S stage, DNA is replicated, right? G2, the cell is going to synthesize all the enzymes and proteins that are necessary to enter the M phase. In the M phase, mitosis stands for the division of the nucleus, right? And another aspect of the M phase is going to be cytokinesis, right? And any cell that is damaged enough to not be divided is going to be committed to programmed cell death or apoptosis. Let's move on. Now, the cell cycle is tightly controlled in our body. The reason why the cell cycle has to be tightly controlled in our body, it's because you do not want cells that either have too many DNA or too little DNA to actually multiply. This is going to cause some misbehavior. Say, for instance, you have a skin cut and then your skin cells are replacing dead cells, and some of the cells don't have the exact same genetic information. Well, at the end of the day, they're not going to behave the same, and that can cause rather difficult situations, right? So the cell doesn't want that. So it's always checking if it is worth maintaining the cell, moving forward with the job that the cell is designed to do, with the cell cycle moving to the next stage of the cell cycle, or destroying that cell. Now, there are three major checkpoints where the cell is checking the integrity. We have the G1 checkpoint, the G2 checkpoint, and the M checkpoint. In the G1 checkpoint, the cell is going to verify that DNA is not damaged. If it is damaged to the point that repair cannot fix the issue, then that cell is going to undergo apoptosis, right? Now, if DNA is okay or repairable, first of all, the DNA is going to be repaired, and then the cell is going to commit to to enter the S phase, right? Then, after the S phase, a very important piece is that the cell has to make sure that the DNA was properly replicated. If the DNA was not properly replicated, well, we have to stop everything, right? So this is the G2 checkpoint, prepares the cell to enter confidently in the M phase, or else that cell is going to undergo apoptosis. What can cause this? It's, for instance, DNA was replicated, but there were a bunch of mistakes, kind of mistakes that are actually compromising integrity of the cell. Therefore, that cell has to undergo apoptosis. And then we have the M checkpoint or mitosis checkpoint. Now, that M checkpoint, it's a checkpoint where the cell is verifying that all the centromeres have a mitotic spindle attached to them, because the mitotic spindles, as they attach to the centromeres, are the one that are going to allow an equal distribution of the genetic information in the two future daughter cells. All right? If the mitotic spindle are not attached to every single centromer, then the cell is going to take the time to allow this to happen. Or if this is not happening, the cell is going to commit to apoptosis. All right? Now, this is basically the internal control of the cell cycle. But then we have an external control of the cell cycle. That external control involves hormones and growth factors. So sometimes you have a neighboring cell that is going to send a signaling molecule. That signaling molecule is going to trigger cell division, right? And that signaling molecule is going to trigger cell division through a process that we call cell communication. And that cell communication is going to lead to a cascade of events inside of the cell that we call the transduction pathway. That transduction pathway ultimately is going to trigger some genes to be expressed or some genes to not be expressed, right? All right, so here we have actually typically what would happen in an external control of the cell cycle. So you have one cell that we're going to call cell A. It's going to release a chemical that we call signaling molecule. That signaling molecule is going to bind on a receptor. Upon binding of the signaling molecule on a receptor, that receptor is going to be activated and is going to stimulate or to activate a cascade of event inside of the cell that we call the signal transduction pathway. That signal transduction pathway is going to either stimulate expression of some genes involved in causing the cells to divide. These cells that are causing this, these genes, which expression is causing the cells to divide, are called proto-oncogenes. Proto-oncogenes. So proto-oncogenes. These are the genes which expression is going to stimulate cell division, while some other genes are called tumor suppressor genes. Genes. Tumor suppressor genes. So tumor suppressor genes stop cell division or trigger apoptosis, while the proto-oncogenes are stimulating cell division, right? So these two groups of genes actually work toward opposite direction, right? During the control of the cell cycle at the checkpoint, the tumor suppressor genes are going to say, well, you know what? This cell is damaged enough. We need that cell to undergo apoptosis. The tumor suppressor genes are going to cause the cells to die. all right? While the proto-oncogenes are going to cause the cell to move on with the cell cycle, all right? I want you to remember that. Now, let me mention that these proto-oncogenes and tumor suppressor genes that control the cell cycle are essential, play a crucial role for cancer, because if a proto-oncogene is hyperactive, you're going to have cell division that is going to happen in an uncontrolled manner. That is cancer, right? By the same token, if the tumor suppressor genes are inactivated so that they can never say stop to the cell cycle even when a cell is damaged, this is going to lead to uncontrolled cell division, which is cancer. So all the cancers we know of are all associated either with a tumor suppressor genes that is inactivated, or a proto-oncogene that is too much activated, and at that point we don't call it proto-oncogene anymore, we call it an oncogene. Let me close that parenthesis. Let's move on. So mitosis, it's for growth of our body. Think of when you were a little kid and now you are an adult. You've grown, right? That growth is supported by mitosis. Wound healing. So here we have a wound that takes time to heal, right? For that process to happen, you need mitosis. The development, think of a sperm and an egg that come together, form a zygote. That zygote multiplies and forms an embryo, and that embryo is going to further develop to form a fetus, and that fetus is going to develop into a perfect individual until birth. And after birth, it's going to be now growth, right? Some development also occurs there. Anyway, then we have tissue repair and replacement of old cells. This is the reason why mitosis is important in our system. Now, let's take a look at the structure of chromosomes during mitosis. Chromosomes, again, are DNA and proteins. These DNA and proteins are so compact, when stained, we can see them under a microscope. However, when DNA is not compact, right, it's rather loose, we don't call this chromosomes, we call this chromatin. All right? Now, during mitosis, each chromosome is going to replicate, right? The two identical DNA molecules are going to be held together through a centromer, right? That centromer is what is going to hold the two copies of the DNA that now we call sister chromatids together, right? So here we have one chromosome, right? We have a centromer area here. After replication, we have one copy of the chromosome and two copies of the same chromosome. The chromosome is xeroxed. They are held together by a centromer. Each chromosome is not called chromosome anymore. We call them sister chromatids, so that based on the name we are giving to the genetic information, we can tell which stage of the cell cycle we are. When we call chromosome chromatin, we know that we are in the interphase. Okay? When we call chromosomes, we know that we are at the stage of the cell cycle where chromatin is condensed, right? When we call the chromosome sister chromatids, we know that the chromosomes have been replicated, right? And that the two sister chromatids are held together by a centromer, all right? And during cell division, now the two sister chromatids are going to go apart, one going toward an opposite direction. All right. The centrosomes are going to be essential for the development of the mitotic spindle. This mitotic spindle are nothing but microtubules that are part of the cytoskeleton. The centrosome is the microtubule organizing center in the cell. And from there, you're going to have a structure that we call an aster. That aster looks like a shining star, right? And that array of microtubule, it's basically happening at the centrosome area, right? Which during cell division occur toward opposite directions, okay? And the centrioles are going to be some short cylinders of microtubules. This is where the building blocks of microtubules are going to come from, right? And develop so that the mitotic spindle can develop, right? Without centrioles, you're not going to have the formation of the spindle fibers. All right, so here we have the centrioles, right? You have the centrosome, which is the area, right? You have the aster, look like a star, shining star, right? That's the aster, okay? And we have the mitotic spindle. And again, all of this, it's nothing but microtubules. These microtubules are targeting something very specific. It's the centromers. And the centromers are actually composed of proteins. These proteins are called kinetochores, but that's another story. I'm not going to bug you with that, okay? Know that the centrioles are going to move toward opposite poles, so that you have one pair of centrioles here and another pair of centrioles here. At some point, the mitotic spindle will start shrinking toward opposite direction. And that shrinking toward opposite direction is going to force the chromosomes to sit toward the very middle of the cell on the equator line. And this is specific to a particular stage of the cell cycle that we call metaphase, right? Meta means middle, phase, it's a phase. The phase during which all chromosomes sit on the equator line of the cell. All right, so mitosis occurs typically on somatic cells, and somatic cells are diploid cells, right? They copy their genetic information, the cell grows, the cell is repaired, and the cell is going to enter mitosis, or the division of the nucleus. And that division of the nucleus is what we are going to focus on right now. We have a prophase, metaphase, anaphase, and telophase. But what specifically happened during these phases? Well, during these phases, during the prophase, the genetic information is going to condense, right? Here, a model cell that we have here, it's going to be a cell where we are only going to have, for the sake of representation, 2N chromosome as 4, right? 2N chromosomes at 4, so that we can track what is happening with the chromosomes. So at interphase, DNA is replicated. And when DNA is replicated, we have sister chromatids for each of the 4 chromosomes, so that if we were to count the number of molecules of DNA, we don't have 4 molecules of DNA after the S phase. We have 8 molecules of DNA after the interphase. Then what happens is that the genetic information is going to condense. That genetic information that is going to condense is going to allow us to see it under a microscope after staining it, right? Then the sister chromatids are going to separate, they're going to part ways. As they part ways, each new daughter cell is going to have 2 N chromosomes or 4 chromosomes with the exact same not only amount of genetic information, but also the same quality of genetic information, theoretically, right? So during mitosis, we go from a parent cell containing 2 N chromosomes to 2 new daughter cells containing 2 N chromosomes. The cells are genetically identical. All right. During prophase, the chromosome is going to condense, it becomes visible, the nuclear envelope is going to be completely disorganized and fragmented. The nucleolus, which is a dense part of the nucleus, right, is going to disappear. The centrosomes move toward opposite ways, and the spindle fibers appear and attach to centromeres, which is going to allow the distribution of sister chromatids toward opposite direction. So here we are in prophase, as you can see. So the chromosomes are visible, right, under a microscope. Okay, so this is a micrograph right here. This is a micrograph. In that micrograph, you can see the chromosome, you can see the aster right here, right? Okay, so you see the aster right here, another one right here, and the chromosomes are more and more organized because the mitotic spindle are now attached to centromeres, right? All right, and the chromosomes are starting to line up toward the equator line, and you can see, kind of how they are starting to line up. And now that leads us to the next step, which is metaphase. During metaphase, the mitotic spindles are completely formed. They are completely attached to the centromeres, right? And they start pulling toward opposite direction. This one pull this way, this one pull this way, so that they are going to line up on the equator line. And as they line up on the equator line, they keep pulling toward opposite direction. They pull so much that the 2 sister chromatids are going to part ways. All right? Okay? But before that, they are sitting on the equator line or meta in the middle. All right? Now, when the sister chromatid part away from each other, we are in the anaphase. Okay, the sister chromatids are now completely separated. You can see this is a micrograph here. If we draw a line here, so you have some sister chromatids on this side now and sister chromatids on this side. Now at that point, they are completely separated. So now they are chromosomes, right? Okay, and now we also have initiation of what is going to become the 2 new daughter cells, right? The 2 poles are completely on opposite side here. We have an indent right here, okay? What we call the cleavage furrow, which is an invagination of the plasma membrane. And that invagination is made possible because of actin filaments, right? While the microtubules are responsible for pulling the chromosomes apart or sister chromatids apart, right? And now we go into telophase and cytokinesis. In telophase and cytokinesis, two things happen. One, it's that the actin filaments are going to keep forming that cleavage furrow that actually bring the cells more and more apart. The chromosomes are now organized in opposite pole, and the nuclear envelope start reforming around the genetic information. Eventually, that nuclear envelope will completely form and surround the genetic information. At the same time, though, the cytoplasm is going to be distributed so that this future daughter cell and this future daughter cell, in terms of quality, they are going to receive all the organelles that are needed for these cells to do the job they are supposed to do in the body, right? So going forward, we're going to have the cleavage furrow that is going to pinch to come more and more together until it pinches off, which means that the plasma membrane has to grow, right? The plasma membrane has to grow. All right, so basically now we have two brand new daughter cells. They are completely genetically identical, and the cell cycle goes on and on. That's how we grow. This is how we replace old cells. This is how we repair the damages that occur in our body. And now we are ready to dive into another type of cell division, that is meiosis. But for now, let's stop our discussion here. Thank you for your attention.